So thank you everybody for joining us today for the one more program of Oncology Beyond the Obvious. Dr. Amish, Vora and myself as program directors are delighted to bring you a totally different publication today. So far we have been talking about phase 3 randomized trials, prospective data, but this time we decided to select something which was closer to home. And we are really proud to be presenting and discussing today a publication that involves molecular data from all across India. And I think it is the right time to discuss this because there are several open questions. There are several incomplete understanding among our oncology colleagues. And there is a potential to benefit from several experts as well as molecular oncology tumor boards. So even though this is a cohort data collected retrospectively, we felt that it was of sufficient value to be discussed in Oncology Beyond the Obvious because it has total relevance to what is the current situation in India. So with these few words, I hand over to Dr. Amish Vora for his comments and the next part of the program.
Thank you so much, sir. It's an honor that we are again meeting. Last month we had the first hybrid OBTO conference where we met physically in Mumbai, and I think one of the best I have attended in last few years, where we discussed a lot. And I'm sure we will continue physical meeting from time to time. But till that time, every month, second Tuesday, we will continue with webinar of OBTO, that is Oncology Beyond the Obvious. Can I have my first slide, please? Okay, next slide. So as Parikh sir said, that OBTO platform today is very proud to discuss this paper. And in the hybrid conference, we discussed a few of the Indian papers. But in the webinar platform, this is our first Indian paper by Indian authors, contributed by Indian authors, and it is published in one of the most reputed journals. The title today, what we are discussing, is Clinical Benefit of Comprehensive Genomic Profiling for Advanced Cancers in India. I would like to take a little time and read out each and every author, because it gives such an immense pleasure to read about this. So Dr. Aju Matthew, Dr. Serena Joseph, Dr. Jeffrey Bobby, Dr. Steve Benny, Dr. Janish Vidu, Dr. Senthil Rajappa, Dr. Nitesh Rohadki, Dr. Bhavna Sirohi, Dr. Ritu Jain, Dr. Vivek Agarwala, Dr. Deepak Kumar Shukla, Dr. Anurag Mehta, Dr. Raja Pramanik, Dr. Vineet Talwar, Dr. Vinayak Makka, and Dr. Nirmal Raut. We want to congratulate all of you for having this wonderful publication and starting the path where we all believe oncology is headed towards. Next slide, please. So the key objective of this study was to study the clinical benefit of comprehensive genomic profiling in advanced cancers in India. Next slide, please. From 12 medical oncologists across India in an anonymized way, 297 reports were collected of CGP, out of which already the known actionable target mutations were excluded. So for example, lung cancer with EGFR ALK, PIK3 mutation in breast, breast cancer with HER2, melanoma with BRAF, lung with BRAF, lung with MET, lung with RET, and so forth and so on. Whenever there is an established actionable mutations, those reports were excluded. So the remaining, there were 221 CGP reports which were taken into this study. This was, again, as I said, published in JCO in 2022. Next slide, please. These were the little bit of characteristics of the study and the patients, equal distribution of male and female. There were 20 liquid biopsy reports also were included apart from 201 tissue. And as you can see the list, almost every cancer in the body was included. Next slide, please. So these are the various platforms and the various laboratories which were used for this CGP. The number of genes which range from 50 to 688, and the type of genetic aberrations which are listed here. Everyone who's attending this, we have sent the full detailed paper on a WhatsApp group of all the oncologists what we have. So you all should have this full paper. Next slide, please. So this was basically the details of the patient who received genomic directed therapy. And if you can see, patient number 13 had benefit of 36 months. This was a cholangiocarcinoma. And patient number 14 had the benefit of 12 months. Remaining all are less than 12 months. Next slide, please. These were the common targets that were used for the treatment. And I'm sure Dr. Sewanti will discuss this further in her panel. Next slide, please. Patients, why many of the patients did not receive the genomic directed therapy in spite of having the target. These are the various reasons which is written. I'm not going to read out each and every, but they have made an attempt to inquire every medical oncologist that you had the mutation, you had the target, why did you not give? And there are various reasons written here. Next slide, please. So the final conclusion again, based on this study, I can see Dr. Ajju Matthew, the first author, we observed that 10% of patients who underwent testing received a targeted therapy and 4% obtained a clinical benefit, defined as receiving a targeted therapy for at least 6 months. And the relevance was, our findings are unique from the perspective of clinical practice in a low to middle income country. It would inform shared decision-making on the role of such testing in advanced cancers. That is what the conclusion was. Thank you so much. Can I have my presentation shut? So this is the paper what we are discussing. And as the OBTO platform suggests, normally Parikh sir takes the next, but because he is involved, I am going to continue. In the OBTO platform, for half an hour, we have an interview. And after that, for 45 to 50 minutes, we have a panel discussion. Entire 1.5 years, we will be discussing only about this paper and its relevance in India and in day-to-day clinical practice. Today, it's my honor, privilege, pride, happiness, whatever adjectives in English you use, I'm going to interview Dr. Purvish Parikh. And I say this full name because Gujaratis have the habit of talking about father's name in between. And I love to do that, especially when somebody else also is a Gujarati. I can talk for hours together for Parikh sir, but I will try to cut short. And Sewanti, I request you, जितना भी time मैं sir को introduce करने में लेता हूँ, उतना कम time तुम cover कर लेना करके. All of us know Parikh sir from TMH days. At least I knew him from the TMH days. MBBS, MD, and all those things, everybody knows he did it from KEM Hospital. He was in Memorial Sloan Kettering Cancer Hospital also for some time. He was in NHS and he taught me how to survive in NHS. In fact, he was the reason why I had an experience of NHS. Excellent teacher and a great human being. There are few things which I learned. First, he's the reason I did my DM. And why I say that, because I was doing my residency in Tata Hospital. There were no DM at that time. And everyone who was there, they said, 2 साल तू टाटा में कर रहा है यार, तेरे को काहे डिग्री की जरूरत है? And he was the first one. He said, nothing doing, get out. Do whatever you want to do. After 10 years, you will not have a value if you do not have a DM degree. I said, sir, फिर सिखाओ मुझे. So after his clinic at 7 p.m., 7:30 p.m. to 9:30 p.m. every day, I used to go to his house in Worli. He used to teach me half asleep, लेकिन मैं छोड़ता नहीं था. And finally, because of all that effort, of course, my genius always counts, but because of all that effort, I got entrance into the DM and I completed my DM. And today I understand the value of that. Apart from that, one very important thing which I learned from him, and very few professors have that, he loves to give credit. And I again use this adjective very sincerely. He loves to give credit. It is not that कि भाई आपका student है तो credit दो. He will fight for you tooth and nail. If it is your hard work, why are you not getting credit? And I think I remember this every day of my life, that if somebody junior to you has done some work, you have to give that person a credit. And I give that credit to Parikh sir to teach me that, to give credit to others. He can adjust in any environment. And, in oncology, I call him a wholesome oncologist, not even just a medical oncologist. You say that clinical trials, when it started, nobody can teach you better clinical trial than Dr. Purvish Parikh. Right from the word go till the closure of the clinical trial, he's taught. He has himself worked in lab. If he talks of NGS, I can tell you that unless and until he has worked in lab, he will not talk about that. You talk about AI in oncology, Dr. Parikh knows about AI in oncology. Medical legal, we all know, we all have taken his help in medical legal aspects, and now he's specializing in medical legal aspects as well. Also, you see the society, cardio-oncology, geriatric oncology, OBTO, why Dr. Parikh's name is there everywhere? Because he encourages you, he teaches you, and he's there to help you. He said, "मैं हूं ना, आगे बढ़ो करके." I think, this is one of the very important point which we learn from Parikh sir. Publications, I don't want to count. 300, 400, 500 के बाद गिनती बंद हो जाती है. आप कितना काउंट करोगे? You name the space of oncology, and Parikh sir has publications of that. He has taught so many students also officially. He is a PhD himself, PhD teacher. I do not want to go into the details of that. But the last few, things which I always remember, he has taught that few sentences, जैसे वो famous sentences होते हैं ना, नाम तो सुना होगा. Not that I am a Shahrukh Khan, Nikhil is here. But there are few sentences about Parikh sir which I always remember. If you can't change the situation, learn to accept it. There is no point in brooding over, oh, abhi ye ho gaya. I think that is very important. What is difference between argument and discussion? Argument is about who is right, and discussion is about what is right. So always have discussion rather than argument. If you have a problem, first find a solution yourself. Otherwise, just don't say, okay, I have so many problems in life. And last but not the least, I still don't agree with him, but he keeps telling me that no human being is bad. It is the circumstances which are bad or good. I think with these few legendary sentences, sir, thank you so much for establishing OBTO and being my teacher, my mentor, and to many others.
Thank you very much, Amish. I should also add one thing, that every time you used to come home, and along with you, sometimes Dheeraj also used to come. So my younger son would always be, a little bit concerned. And then he would go and tell mom, my wife, Dr. Appana, that now these two will start fighting. So our discussion was considered as fighting by my younger son.
Right. And Kashish today is, one of the pioneer and the founder of Kavina Creations. I think the seamless integration or the seamless implementation of all these OBTOs and many other conferences is done by Kavina Creations. So, ladka bada ho gaya, sir. Okay, so with that, let's start, sir, the discussion about this paper, which I presented slides about. As the tradition goes, sir, it is a rapid fire few questions which I will ask you. And since they are rapid fire, I would request you to have rapid answers also so that we can move on. Sure. When was the first experience of NGS or the CGP, sir?
So you already mentioned it, but I was privileged to do molecular oncology as my thesis from Mumbai University. Okay, so the first time that I did Sanger sequencing was as a part of my thesis while I was at Memorial Sloan Kettering Cancer Center. And then, fortunately, I was given the consultant's job at Tata Hospital. And there, with the help of Anuradha, or I should say, Anuradha did it with my help, we were able to establish molecular oncology lab for Tata Hospital. So it has been a real journey from hands-on work in the lab as well as establishing the lab. So I've seen Sanger sequencing and right up to NGS.
Right. So when we didn't know the word oncology, you already had established a molecular oncology lab. So that is amazing, sir. Since you mentioned about Sanger sequencing, I still get confused sometimes, because when in few of the NGS report, when I read, they say confirm with Sanger sequencing. So I would like to ask you, what is the difference between Sanger sequencing and NGS, sir? Quickly.
So that's a very important question. In Sanger sequencing, and Watson and Crick came up with the double helix DNA before I was born. In 1953, they came up with it. And in 1977, Sanger sequencing came into the picture. So what happens in Sanger sequencing is there is a DNA strand, and then that DNA strand as a duplicate is made of the DNA strand. So extension using a primer is made. And in order to find out what are the bases, the test is carried out in 4 separate tubes. Okay. As you know, the structure of the DNA, we have nucleotides, we have the sugar base, the hydroxy, and the ATP. Whenever there is a hydroxy molecule, then that 3' continues to be extending. So you make a copy. Now, in this 4 tubes, one contains A, one contains T, one contains G, and one contains C. That are the 4 nucleotides. One of the nucleotide in a small portion is D-deoxynucleotide. That means the hydroxy molecule is removed from it. As a result, whenever that molecule gets incorporated into the duplication, the termination occurs of the sequence. So the principle of Sanger sequencing is primer extension and termination. And the termination of the D-deoxynucleotide is with a radioactive attached to that nucleotide. So then when you put the samples in 4 different columns that we did in a vertical, then they would move forward according to the size. The movement of the smaller fragments will be much more. And then when you put it close to an X-ray film and develop it, you will see the sequence or the movements of the fragments in 4 different columns. All the fragments in first column would be A, all the fragments in the second column will be T, G, and C. And then you put them all together to find the sequence. So this is a very time-consuming, expensive method and very slow method of doing it. But because there is no amplification, so the chances of a mistake or error are less. Therefore, Sanger sequencing even today is considered as the gold standard. If any of the other methodology does provide something which is of doubt, we have to go back to Sanger sequencing to verify. And the critical thing is there is no amplification, and therefore, so the error is also not amplified. So this is Sanger sequencing.
Excellent, sir. So this is something which is cleared. Just wanted to, sir, know before we go on to the number of genes and how to read NGS report. Tissue is an issue. Always, we have given, all of us have given lectures in some platform. Tissue is an issue. Since we are talking of this paper, which has a relevance of utility of NGS, I wanted to ask you a few basic questions, which can it impact the way NGS report comes. So my first question is that, does tissue become an issue if not properly stored and not properly transported? Or is there a difference between a fresh tissue NGS and FFPE tissue NGS?
So whenever you look at NGS or any sequencing, we have to start with the DNA, or we have to start with RNA and then convert it into a cDNA. So the entire process, outcome and the success will depend on the integrity of the DNA. If the DNA is not preserved, or if the DNA is not available, for example, if there are only 5% malignant cells in the tissue that is provided, or if it is in the FFP blocks in the last 10 years and it was stored at room temperature in Rajasthan, where the summer temperature is 43, then obviously you cannot expect the DNA to be intact, and therefore what is the outcome of the NGS would be of questionable. But I should also like to point out that since liquid biopsy is of great value and its utility is increasing, I must give you an example of a non-oncology condition. When a elderly mother is pregnant and we want to find out whether the baby might have Down syndrome or not, today we do not have to put a needle in the amniotic fluid. It is the mother's maternal peripheral blood that is evaluated, and the child's circulating DNA is identified from maternal blood. So this is how proficient, how sophisticated the technique has become. So the tissue being the issue is going to go down as we progress further for any prospective study. For a retrospective study, of course, because the sample was collected and stored a long time ago, that could still be an issue.
That could still be an issue. So tissue can be an issue, and in future, liquid biopsy probably will take over that. Second question is, sir, I showed you a slide where there were 9 to 10 different labs were used. And in some NGS or in some CGP report, they have used 52 genes, and some it is 688 genes. What is the significance of these number of genes, sir? Is there a minimum criteria, or are there any standardized guidelines for selecting the number of genes? Can that impact also the report?
You have really put the most important question in the front, and that is, what is the definition of comprehensive genomic profile? On the one extreme, we have evaluation for single genes like EGFR mutation. At the other extreme is the Human Genome Project, that is comprehensive genomic profiling. And that can be expensive, that can take a long time. The first human genome was sequenced over 10 years and costed $3 billion. Now everybody knows today for an individual, the whole genome sequencing is possible in 4 hours also. So the technology has advanced, the cost has come down. But between these two extremes, there are lots of gray areas, and one of them is the uncertain definition of comprehensive genomic profile. So we commonly use the word comparing apples to oranges, but when we have to compare a test of less than 60 genes with a test of more than 600 genes, it is not comparing apples to oranges, but it is comparing a grape to a watermelon, because the size is so different.
So I get your point. So basically the tissue can be an issue, the number of genes can be an issue. Also, sir, please tell me, there's a lot of these lab people, every second day somebody opens up a lab doing CGP. And they say कि सर हम तो RNA भी करते हैं, हम तो whole genome sequencing भी करते हैं, आजकल हम WES whole exome sequencing भी करते हैं. What I understood that if I don't know what I'm looking for, and if I order whole genome sequencing, the depth I'm missing out on that, and that can impact the report. Am I right on that, sir?
So you are very much right, but it not only depends on whether you're doing whole exome sequencing or comprehensive genomic profiling. It also depends on what the equipment and what chemistry you are using in NGS. For instance, Applied Biosciences, Iron Torrent, and there are 3 other, Roche and other. Each one uses a different chemistry to do the processing. And for the limitations of the length of the DNA that is being replicated is also highly variable. So it could be very small for Sanger sequencing, and it keeps on going up. As for instance, if you take Iron Torrent and you say MySeq, HiSeq, and the more latest machine, the frequency, the read length, and the speed increases. So it is not just the lab itself, but it is also the equipment that they use, the chemistry that they are using. And please understand that whether it is comprehensive genomic profiling or whole genome, as far as NGS is concerned, besides the sample collection, processing, and transport, it is the wet lab, what we do in the wet lab, and even more importantly, what happens with the bioinformatics, that could be highly variable. For instance, one lab will say that 1% change is sufficient to call it a mutation. Another lab will say, no, 1% is not enough, it has to be 3%. So that variation is also.
Sir, when you say so much of technical word, it reminds me one of the days I got a call from a urologist who had attended one of my lecture and said कि अमिश ये HRPC में मैंने docetaxel शुरू कर दिया है, कैसे देना तू बता. So I think at NGS we are probably, at least I am at that level. That is, I must confess. So that comes to one of the important point which I face every day is how do I read my CGP report, sir? Because that also makes a difference. I'm extremely lucky that I have Dr. Amit Verma. But if I, if I take out Dr. Amit Verma from my equation, like, just wanted to ask, day before yesterday, Dr. Navdeep in one of the groups said that, RAS is wild type in colon cancer, but there is a PIK3CA mutation. Will it impact anti-EGFR therapy? And I had read that earlier for one of our patients, and it said exon 20 PIK3CA mutation, if it is there, cetuximab and panitumumab may not work. If I don't know that, sir, and I just see the RAS wild type, I give cetuximab does not work, and I say my CGP failed to help patient. Is my understanding correct? Are we naive in reading the report of CGP today, sir?
So, CGP is quite different from looking at a PET scan or a CT scan. There, we can clearly identify where the abnormality is, what is the description of that, and whether it's benign or malignant. It's very easy to make up. As far as CGP is concerned, most of our colleagues who are not conversant with terminology would depend on the lab report. The lab report will be limited to what they are able to understand and what is there in literature.
Correct.
But the most important concern that I have is that no lab or hardly any lab is willing to give the raw bioinformatics data to us. Why is it happening like that? If that bioinformatics raw data comes to us, then we can use publicly available software and databases to actually interrogate and find out what exactly is the abnormality. And it is the molecular oncology tumor boards like what Sewanti does and Bhavna does and Amit does, that actually help in better understanding beyond what is written in the lab report. And like you rightly pointed out, there are two things. One is that if there are 2 or 3 changes, which one takes precedence over the other?
Correct.
The other is that if there is a VUS, something that we have identified today, which we are not sure what it is, the publicly available database continues to have more and more information coming in all the time. So, for instance, MSH3 mutation in chromosome 5 was seen in one of my patients who was worried because they had family history of malignancy in 6 blood relatives. The first report was done in August 2021, at which time it was VUS. Now they came for follow-up, and I was able to look at the publicly available database and interrogate it. And today it is clear that that is no longer VUS; it is a benign alteration. So imagine the relief that we can provide to the patient, because that fear of being at high risk for cancer is gone. So therefore, we must always look beyond what is the report that is provided by the labs.
Right, sir. So, you have given an extremely important example, sir. You send this report to me and Sewanti, and assuming if the patient with cancer would have been there, whatever I read, MSH3 mutation, MSH3, and MSH2 work side by side, and they are part of a DNA repair mismatch. If I don't have the knowledge, probably I would have offered immunotherapy to that patient. I called up Dr. Amit Verma. Amit said, no, wait. You have to see in totality. It can be a VUS, it can turn into benign, and now you are saying benign. So one report you reported as benign, I would have reported as MSI high, and probably I would have offered immunotherapy. So this is where we are, and this is where my biggest worry, sir, is. But anyway, we'll move forward because we've already taken 15 minutes. So I will not right now ask you the last question, which was I'm tempted to, maybe you can write in the chat box, why Shiva trial failed. because we have another 15 minutes to discuss about the paper what we are discussing today. So, sir, you in your introduction, you have already answered the importance of this paper, so I'm not going to ask you about that. Let's jump to our second question. We always call this OBTO as a practice changing paper discussion. So the conclusion was of this paper, and I just want right now yes and no answer, and then again I will ask you the same question in the end. The conclusion was you should be judicious in offering or ordering CGP in your patient. Sir, yes or no?
So first, let me start with a disclaimer that I'm playing the devil's advocate. Okay, the publication is useful. The publication has a lot of evidence in that, and there are some good lessons that we can take home. But I feel the conclusion is exactly opposite of what should be. I feel that the number of patients who benefit is more if we pay attention, if we are appropriate in our application of the knowledge that is currently available.
Perfect, sir. So, this was the methodology, or the title was multi-center retrospective cohort study where 12 medical oncologists have, contributed. From this perspective, sir, you are comfortable in accepting this data, right? I just wanted to have just the title.
So, retrospective study is real world data.
Right.
This was samples that were sent prospectively, even though the data was analyzed retrospectively. Okay. And it is a cohort of patients, but the cohort number is sufficient to give us some indication of what is happening. And the data that has been there, you have already shown how many genes were involved, how many cancers were involved. So it is interpretation, and my conclusion is going to be different from what it is there.
Understood, sir. Perfect. The primary endpoint of the study was, basically eventually received targeted treatment. That was the primary endpoint. And the secondary endpoint was, this is something unique, sir. People who have taken more than 6 months and people who have taken more than 12 months. Now, what is your comment on these primary and secondary endpoints, sir?
So, coming to the second part first, that is the response, the meaning of the response. I have never seen a trial where the definition of response is the number of days or months or years that the patient has been on treatment. Okay. The response is always evaluated by RECIST criteria on imaging or on the question of biomarkers, if it is, for instance, CA125 in ovarian cancer. Though these are surrogate markers, these are known, well-established surrogate markers. Number of days or months for which the treatment has been taken, I don't think that is a surrogate in any case. For instance, let us look at the real-world circumstances in India. The patient is being treated by me. 3 months later, he feels that I'm not giving him enough attention. He goes to you, Amish. You will continue the treatment because he is responding. But as far as this retrospective study is concerned, they will say that the patient has not responded. So these are some things that we could have. The other problem that I have in the methodology is what is the meaning of the inclusion criteria? At one place, it is written that all cancers, all stages are included. At the other, at the end, in the conclusion, also elsewhere, it is written that this is only for advanced cancer. And there is no definition of what advanced cancer means in the publication. So again, this comparison of apples and oranges comes into the picture.
Before I move forward, Dr. Arju Matthew, please understand that this is one of the important paper on which we are discussing. We are not trying to negate or anything. It is just that our perspective of understanding of this paper, and Sewanti will go further details into it. You have all the reason to put your comments on the chat box also. Okay. So coming to the results, sir. I'll read out the results, what I have written. 297 reports received, 221 included, 76 were excluded. Now, I don't know the reason why they were excluded. I don't think that should have been done. 43%, that is 96 out of 221, had a targeted treatment option. 21 out of 96 received targeted treatment. They have given the reason why so many others have not received, but again, this is little off. 33%, 1/3 of patients restricted themselves only to HER2 exon and EGFR. Looks like as if there were not many other targets, which again is something, little different from what we see. 17 out of 221 received targeted treatment. 9 out of 221 benefited. 2 out of 221 are exceptional responders. Now, this result somehow, sir, when I look at my personal experience or international publication, doesn't look very near to the real practice. But I would like to know your thought process on these results, sir.
So first on the exclusion. Okay. This is one of the few papers where the attempt has been made to reduce the numerator. Okay. And if that is there, it is fine. But then the title has to be different. It should be that comprehensive genomic profiling after deducting or after subtracting common mutations with single gene or limited gene panel, it did not benefit. Okay. When you say CGP did not benefit, does it mean that exon 20 mutation is not picked up by CGP? Of course, it is picked up by CGP. So that gives the title gives a misleading idea, and therefore the 297 and 2 contentious issue. The other contentious issue is there were several instances where the patient had a mutation but did not get the drug that was available to target that mutation. Patient general condition was bad, patient refused, patient did not enter a trial which was available. The treating doctor felt, no, the report may be okay, but I don't want to treat him with that. How can we say that these reasons are valid enough to analyze and interpret that CGP is useless? CGP was good. CGP identified the patient. But for subsequent reason, whatever may be the reason, the patient did not get the intended treatment. And that is not the fault of the CGP. It is the fault of, if I don't want to use the report to treat, to guide the patient's treatment, I have no business to send the sample in the first instance.
Right, sir. So we have just 2 more minutes left. In fact, 1.5 minutes left before we give mic to Dr. Sewanti. My point was 2. One is, before you say any parting comments on this paper, should PDL1 be a part of NGS going forward for every patient of ours? Maybe you or later on Dr. Sewanti also can take. And sir, any parting comments on this paper?
So PDL1 is immensely useful. And I have seen patients who had failed 2 or 3 lines of treatment, and then with the immuno-oncology drug, they are alive for 5, 6, 7 years. In fact, one of my colleagues, Dr. Lalit Mohan Sharma, shared a patient who was off treatment for 3 years and is incomplete response. So I would definitely include PDL1 testing, even though there are some limitations. What is the cutoff? Is it 1%? Is it 5%? It is 49%. There are some molecules that are marketed irrespective of PDL1 status. Having said that, I would still do PDL1 in all patients who are able to afford that sort of treatment. And my parting comment is that this publication is very valuable. It is a great idea that 12 medical oncologists have got together and pooled their data. I was personally able to speak to at least half of them and understand more about what they have done with the patients, which may or may not be included in the study. But I feel that the methodology has several limitations. The title is misleading. And the way that the 297 patients have been divided, as well as the way they have defined response, is something that I would like to question. And therefore, everybody should be aware of both sides of the coin. Thank you.
I completely agree, sir. Thank you so much. Aju, I checked at my 50 last CGP patients, and it's almost 14 to 15% are very good responders based on your definition, if not more. And so with that, thank you so much, Parikh, sir. I think this, again, it reminds me of the days, if people who are aware, Tata Hospital, mein 7th floor NX building was medical oncology. And every afternoon, Parikh sir, or thrice a week, Parikh sir would come to teach us. We were interested in his lecture, of course, but we were also interested in pizzas and pastas, what he used to get for all of us. It reminds me of those days, sir. And with that, sir, I would request you to introduce our moderator and take it forward from here.
Thank you very much, Amish, for, you were the interviewer. You could have pulled my leg in many ways, but you were kind enough to me, so thanks very much. It is now my proud privilege to introduce to you Dr. Sewanti Limaye. She actually is very well known, especially in the Bollywood industry. Any star who has a cancer would first go to her for advice and treatment. She deserves this because she has got cutting edge training, education, and qualification from United States. And she has also set up a molecular oncology tumor board. Some of our colleagues felt that that was conflict of interest. In fact, we feel that that makes her qualified to moderate the discussion, and of course the panelists are always there to question or to give their opinion, so we expect a very balanced view. The panelists along with me are Dr. Vinit Govinda Gupta. I'll not waste much time in introducing everybody. Dr. Nikhil Gadial Patil, Dr. Randeep Singh, Dr. Aju Matthew was the first author of this paper, Dr. Vamshi Krishna, Dr. Anuradha Chogle, Dr. Raj Kumar, and I think we should be very grateful that he is such a senior person in molecular oncology is here. And therefore, and of course, Dr. Suruchi Agarwal from MedGenome. So with this, I hand over to Dr. Sewanti Limaye for the discussion.
Good evening. Thank you as well, and I will just by saying as
we can't hear you well. Your volume is low. Sewanti, we can't hear you well.
Moderator without a voice.
Sewanti, what you can do is you can join in from your phone also and keep both phone and computer on. Probably that would serve the purpose.
So
That's now perfect.
Okay. All right. I was just saying a moderator without the voice. That wouldn't work. Okay. So let me scoot into this. And so first of all, very grateful for this opportunity, and echoing Amish's thoughts about Sir being phenomenal and a pioneer, and really bringing to the fore the concept of molecular oncology, and the first lessons learned were from him. And Amish, just a line regarding the voice of molecular oncology. And in parallel, standing tall with Sir to bring this oncology beyond the obvious. And that voice is loud and clear, and hats off. So it's an honor to be discussing today's paper along with the esteemed panelists, and complete acknowledgement to the conflict of interest that Sir just mentioned. And I was just telling Sir that that is a conflict that remains with me. It's written on my forehead now. So I can't run away from that. Wherever I go, I'm running MTVs. So that's an honest confession. But I hopefully would be able to shed some light along with my panelists onto the wonderful manuscript. And so I acknowledge the fact that this is a landmark paper. This was a seminal attempt, and kudos to Dr. Aju Matthew and team, and Nirmal Rao, my dear friend, to have put together such a compilation and have the courage and the audacity to publish it in a world-class journal. So that is a 2 thumbs up from a very humble person. Having said that, of course, when a paper makes it so tall and clear, it will achieve all the attention that you are achieving today. And that is what we are here to discuss. And that was just a disclaimer, a background from all of us here. And I'm sure all my panelists will concur. With that, I will open the floor to the discussion and we'll just present some of the storyline to then bring you to a conversation regarding the paper. So is it okay if I share my slides?
Sure.
So I begin by what is being discussed. When we think of precision, and I begin with precision because we utilize different methods of testing as a part of precision, as a part of treatment of our patients. And we know that there are subtle differences between personalized therapy and precision oncology. But largely speaking, there are concepts to identify a certain type of paradigm. So question open to the panelists through this entire list of things that are written on the slide. What do you think encompasses precision, the single gene finding, IHC, hotspot, gene expression analysis, chemosensitivity, tissue or liquid biopsy? Is it all of the above? Or is it targeted therapy, hormone therapy, immunotherapy, or a combination of all, or chemotherapy for that matter? And for that, I'll come to none other than to begin with the leader.
So I think as far as precision is concerned, it is, giving the best efficacy for the least amount of toxicity to a patient. And therefore, in my opinion, it includes everything that you have put on the slide. It could probably be more than this as well.
Wonderfully said, sir. So that was the goal, to drive home the fact that everything is game. When you are treating the patient, and if you have the goal of getting the most benefit, the best outcome with least toxicity and most preserved quality of life. In that process, of course, there are hits and misses, and we are all learning, and the technologies are evolving. With that, the next thought. What is CGP for you? And for that, I'll come to Nikhil. Nikhil, your thoughts on what would you think of comprehensive genomic profiling, and do you order them? Do you order them frequently? And why do you order them if you do?
So when you talk about the CGP, it is a kind of an NGS testing which tries to look into, let's say, hundreds of genes, or I would say multifold of genes. And the reason we want to have that information with us is just to gain some kind of understanding about how we are going to treat the patient, especially when we are looking for some newer therapeutic approaches, or we are also looking for sometimes some clinical trials, or sometimes whether we have some drugs in the pipeline, whether we can have that kind of treatment available for the patient. And this is usually, I do not do upfront for most of the patients, but probably I would normally keep it somewhere at the back end, where I would like to utilize this particular CGP for especially for the patients where the options for treatment are quite limited, and so that we can try and explore some more or newer approach in terms of how frequently. I wouldn't say very frequently, but at the same time, we do have some, just to give some ballpark figure, maybe in my practice, maybe I would say less than 5% of patients may undergo CGP.
Thank you. Thank you for sharing that very relevant and important piece of information and your style of practice. And these are the variations that we will see as we interview our panelists. And I will ask the similar question to Dr. Vineet Govind Gupta. Vineet, what are your thoughts? And just leading to the next segue, are all CGPs the same? Do you order different CGPs, and what is your take on that?
So once there is a, to some level, we have to look for biomarkers in every cancer. The only question actually is how big the panel is, meaning how wide a net you are casting. Are you just looking for a certain smaller panel of biomarkers, or you're looking for a larger panel, including biomarkers which might be there in very rare patient, maybe 1%, 0.5%, and so on. So when I think about CGP as it is being discussed today, or it was discussed in the study by which was discussed earlier, we are mainly dealing with the wide net. We are mainly dealing with the analysis of large number of biomarkers beyond what is considered as standard. So definitely all CGP is not the same. It will depend upon the quality parameters, and also very importantly, it will depend on what we are looking for in a particular indication. And it will also depend on the type of tumor where we are thinking of implementing the CGP. So definitely there is a nuance to picking the kind of biomarker testing we want to do in a particular cancer type.
Great. So I guess the question that I actually wanted to ask in this, and very well answered, and I'm coming back to you, Dr. Vinny, is when you order a CGP from different labs, do you see variations?
Yeah, definitely. So there is definitely quite a bit of difference in different labs, and that needs to be reconciled, because I do notice some patterns. I don't know what is the data, but there are some patterns of CGP I see across different labs, and you don't get the same kind of report from every lab.
Great. That's exactly the point that I was wanting to drive home to the discussion, that CGPs and CGPs and CGPs, and whether you talk about gold standard labs or non-gold standard labs, and who knows what that means. It's truly variations, and variations that have been widely written about, not just in our country, within the country, but internationally, and with the pioneers of people who have declared themselves as the kings of CGP. So then comes the question, are the reports that are being provided to us enough? Are there any challenges in clinical application? And do you apply the treatment options alone, or do you discuss them in forums? And how do you go about doing that? And for that, I'll come to Vamsi. Vamsi, very interesting question being posed. When you read the report, do you feel comfortable in interpretation? Do you lean on others, try and understand? How do you do it? You do a lot of CGP, I'm aware, and I want to know your style.
So I think the important point to remember is you cannot just go by word to word what they say. You need to check what reason they have given for providing a particular drug. The other challenge is, if the lab is not something that you're very, very comfortable with, it's always good to go into the depth and see what exact mutation they're talking about. Is that really a mutation that is pathogenic? It's not that difficult to find out these. And of course, it's always easy to put it up to a company and ask them that, look, have you gone through the base, and is it really pathogenic? You cannot take any report at face value. That is very, very important. You have to be able to analyze it. If you cannot analyze it, find someone who can do it for you. So don't just rely on the one-person lab who says, oh, this drug can be used in a different setting. There has to be biological plausibility before you use it. And there has to be a reason why you want to use that drug in that particular patient. Now, beyond this, I have to say a philosophical thing that as a pursuit of science, we all would love to do CGPs everywhere, but we have to understand that when we are talking in a bigger audience, the audience should not go away with the imagination that every patient do it, otherwise you're doing something wrong. I think that's the other thing which comes out when you look at this paper also, because you have a group of patients where it will help, there's a group where it doesn't help. Pushing somebody who cannot afford to do the test is something which I think should not be done. So that's just a more, a bit out of the way. This is
great point driven home. Curation of the data, curation of the output on our own, curating the report on our own, taking the time to curate the report on your own, or at least leaning on someone to have as your main, partner in crime to curate the results of a report, I think is very critical today, because I feel I many times don't have the time and the energy to go and look up alteration, and assuming labs interpretation of the tests in today's times is not considered the in thing. The in thing is actually what Dr. Vamsi just said. You need to sit down and look at the alterations and really do your own curation or lean on someone to do the curation. With that, I'll come to Dr. Anuradha and then to Dr. Suruchi Agarwal, and mainly asking them this question: how people lean on them for inspiration. You get random calls, reports, asked what's done with this alteration. What are the issues that are brought to you? I would start with Dr. Anuradha. Adam.
Your voice is cracking.
Okay, I think you may need to share the video. Then get a good.
Sewanti, just repeat your question. I think she didn't hear it.
Okay, my question was, to lean on you for curation of reports. Do they call you and say, okay, what does this alteration mean? Can you please help me figure it out? Whether this is, this means anything? Is this pathogenic? Should I be treating this particular, or is the report correct?
Yeah. So many times so happens some of the labs they don't give an elaborate report, but with foundation one that they give an elaborate report, that is very fantastic. But actually we need to decide as per the tier 1, 2, 3, 4 whether is the driver actionable, whether we can treat it, or whether it's a clinical trial basis or what. Second thing, when you're looking for the report, we need to explain that this report is very giant. For that, you need to look whether the overall coverage is good, whether the patient has, whether the sample has got a good coverage, or whether the VAF is very good, very allele frequency, whether this is a canonical mutation or whether this is a non-canonical mutation. We should make them understand about this report. And of course, we have those interpretation of the reports with the targeted therapies which are on, and most of the time it has been elaborated in the report itself.
Excellent. Thank you. And I'll ask the same question to Dr. Suruchi.
Sure. Hello. Hi, everyone. Thank you for me being on the panel. So on the actionability of the report, I would like to say that there are many aspects in the NGS report that one should look at. So it's not only dependent, like Dr. Vineet also mentioned that first of all, we should be sure about what are we looking for. So what is the phenotype of the patient and how we can correlate the results with the phenotype of the patient. That is one thing. Then as Dr. Anuradha mentioned that there is like variant allele frequency, whether the variant is genuine, what is the coverage, all those things are also mentioned on the report. So actionability, apart from FDA approved therapies for which the variants are there, so that has to be taken into account, like what is the combination of the variants that we have got, or what are other VUS or other additional biomarkers that we have got in the report. So it should not be looked upon in a individuality, but like a combination of all the factors and the medical other things that has to be taken care while interpreting a NGS report.
Great thoughts. I think a lot of this echoes what we have been trying to convey, that there are nuances to reading the report and it's not as straightforward as initially we thought. And there are angles with which we have to interpret. And many times we don't understand it all and we need to lean on our molecular colleagues. And many times we have to discuss it in forums to really get to understand who's weighing where and how would we take a final decision.
Yeah,
so,
I want to add to it. So I think this is very important that when we do a comprehensive genomic profiling, the reports should be discussed. It's not like just if we see a tier 1 variant, or there are other variants also sometimes present. So if we just take a decision on the particular variant that we see at first glance, that is not recommended. We should discuss the report completely and then only actionability should be decided.
Excellent, excellent. So I think,
and again, I would like to cover here, when there are compound mutations and both of them are driver actionable, then it becomes a problem which one to treat. So many a times we have learned from the centuries that whenever there are fusions and when there is a point mutation, then which one is to be treated first? And now it is considered that fusion should be treated first and then the point mutations. So there the confusion arises with the clinicians, which one to take first.
wow. So very nice points brought forward. And now just a quick question to the guru, Dr. Amit Verma. I see he's also joined in. If I may ask you a quick question. So people turn to Amit for a lot of solutions in molecular. And when you have multiple alterations enlisted there as a part of a CGP report, how do you process?
So it's a very difficult question, but I tend to see the VAF frequency. If I see a dominant VAF, I think that should be the first one to be targeted for sure. There's no doubt about it. If there are tandem alterations in the same pathway, definitely it is, you have to try the upstream as well as downstream. It's prudent to do that. If you have multiple pathways at the same time, I try to combine therapies, a combinatorial approach rather than a monotherapy approach. But if not feasible, then I try whatever is by my best, you can say, guess. It's just a pure guess. I'm not the right person to give an objective answer to that. But I try to see that if the dominant pathways are involved, I try to target them first as compared to the rare things. So again, rare thing happens rarely in the cancer, so we have to follow that rule as well. Yeah.
Thank you.
Sewanti, can I ask you a question?
Me.
How important is allele frequency when we decide about the implications of a genetic alteration?
So are you talking about the liquid or the tissue?
Whichever way.
So in liquid, the VAF have no significance. Whatever you see, it is just a minuscule of the information. So you cannot corroborate VAF with any interpretation on that. On the other hand, if it is a tissue, yes, VAF have some, you can say, understanding. If it is a truncal mutation, you will see more VAF. If it is a branch mutation, you will see a less VAF. If the VAF is in the range of 50%, you can assume that it is probably a germline one. If not, then you can guess. If there is a VAF which goes beyond 70-80%, then it means that the loss of heterozygosity has happened. On the other alleles has been lost. So these are 4 possibilities which normally I encounter. But again, it has to be seen holistically, the type of mutations and the pattern of mutations actually.
So again, the guru speaks. So great insights, and with that heavy duty molecular message, I move over to some realizations about the paper. Now, coming back to our paper in question, and the reason why I presented this sort of an intro is to understand what we understand of CGP, what we understand of precision, and what is our main goal. And that's why I started with a question to Parikh sir as to what is it that we are trying to achieve with whatever test we do. What is our main goal? Where are we trying to get to? And with that clarity, how do we get there? In that context, coming to our paper in discussion, I will turn to Dr. Randeep Singh, and I'll ask Randeep, what does he think of the paper? And then my next question would go to Aju, and you can see the slide out here, Aju. So before I come to you, I will ask Dr. Randeep, in the context of the paper that we are discussing or talking around today, and this current slide where there is a difference in actionability that was discussed in the interview with Dr. Parikh before this moderation started. Your thoughts on the paper? What did you take away from this manuscript?
Sewanti, while I was going through the paper, I hope I'm audible well.
Yes. Yes.
So I was going through that paper. I realized 96 out of 221. I just jot down that figure. I wrote that while I was reading the paper on the weekend. 43% positivity. I mean, as Purvish said that apart from the conclusion, if you don't read the conclusion and read the whole paper, 43% positivity of genetic aberrations, and we have negated the standard EGFR, HER2, EGFR in lung, HER2 in breast and others. These are not standards where we are using. These are core CGP reports. These are not FISH amplified and others. 75 who have not got targeted therapy, 20% they had poor performance status. That message which I learned was that yes, we all have started ordering CGP early, provided the patient can afford it and the family is motivated to get the drug from XYZ corner of the world. 30% the trial was possible, but because of the poor access to the clinical trials in India, still whatever we may say, the access to clinical trials is very, very poor in India. It is not about we are not conducting, but we don't get that much trials to our continent. And then 20% other standard of cares were available. So of these 75 patients, I feel another 30 gets added to that 4% group. So what I feel that the figure somewhere will go above 10% if we include these 75 who have not received targeted therapy. The 21 who got targeted therapy, I would add another 15 to it, because these 75, at least 20-30% will be receiving some sort of targeted therapy. Other message which I learned was that yes, HER2 is ubiquitous. It is something like a TMB. You are having HER2. Gradually we started HER2 from breast. We learned lung has a HER2. We learned colon has a HER2. In last 2 years, we learned the importance of HER2 in cholangio and biliary cancers. And you can see in this paper also that there is a clear message that once we have newer monoclonal antibodies such as trastuzumab deruxtecan and others, these CGPs and other HER2 mutations will become much more important, that even minor expression of the HER2 will make a difference for the drugs like TDX. So I feel that yes, 4% is not the right value. As Purvish sir also, I will put this figure somewhere around 10 to 15%. I would have been very happy if the response rate would have been the primary endpoint. It would have been more robust. It would have been more based on imaging as well as clinical impression. PFS is something which is very varied, because there was no standard set guidelines that you have to do a CT scan every 2 months, you have to do a PET scan every 2 months. If I'm starting someone on the last line of therapy, I may just not do the scan. And if I don't have anything, palliative care versus giving trastuzumab for a 4th line cholangiocarcinoma, will just say you keep on taking it till you have a benefit, and that amount may come to 6 months, 12 months, 1 year, or clinically. So I've been happy to see the response rates, and we all believe that if we have a good response rate, 4-5 months of PFS will be achieved. So I think it is a short summary. There are many other points, but I will stop here, Sewanti.
So how did I ask the right question to the right person?
But I think Purvish sir had actually nailed it. Aju, I would be happy to hear from Aju now after these comments, that yes, he would be happy to hear these.
I misspoke. I just want to go to Dr. Rajkumar, one last person before coming to Aju. So I haven't spoken to Dr. Rajkumar yet. Following right after Dr. Randeep, Dr. Rajkumar, your take on the paper. 2 people, just to summarize from the panel. And but I have to say, Randeep, so well summarized. You'd read it so well. I'm not sure if all of us would have read it like that if we were on the panel. So kudos to you. And is Dr. Rajkumar there?
Yeah, thank you, Dr. Sewanti. Can you hear me?
Yes, sir. Yes.
Okay. Let me share my thoughts about first the paper and then also about CGP as well.
Yes.
The first paper, I think this has been discussed threadbare by now. Purvish and Randeep have given their perspectives. And I personally think that while there are these kind of newer tests are likely to enlarge upon the availability or choice of different drugs for specific malignancies based on their mutational profile, we have a long way to go still. Why do I say that? The point simply is that at this, the technologies which we are using, the different gene panels which are being used, everything is so variable. You do not have a commonality with regard to whatever is being done. So trying to compare one with another is going to be difficult. Now, coming to the CGP in itself, I would say that CGP has a role to play, but please bear in mind the limitations as well. Now, in this context, tumor heterogeneity is going to be very, very important. There was a landmark paper in NEJM on renal cell cancers. They actually did a complete genomic and transcriptomic analysis from different parts of the tumor, from different metastatic sites. And what they found was more than 60% of the variants were not detected in all the regions. So which tells you that the sampling which you're going to be processing may not give you the complete profile. And this is particularly true of your tissue biopsies, which you're going to be dealing with. In that context, maybe I am biased, but I personally feel there is a role for the cell-free DNA, because here you are likely to have a much better repertoire of the tumor-derived DNA material to come out. Of course, there are likely to be limitations. People are trying to overcome some of these aspects, but I think that is one situation which could be looked into, and I think people are working towards it. But there is a caveat there. You will need to have a depth of coverage which is going to be 10,000x. If for tumor you need anything more than 1,000, for cell-free DNA you need more than 10,000. So which means you really need to have a good sequencing system, and the companies which are going to be dealing with that will need to factor that as well. And ideally, you would also want to compare, at least in the initial stages when you're trying to set up something, the germline. So you take your peripheral blood mononuclear cell DNA, and then you take your tumor and then your cell-free DNA. That way you will have a clear comparator to say which variants you can exclude as being related to germline and not likely to be pathogenic. Now, coming to the pathogenic mutations, one of the things which Purvish did mention to me before in the process of asking me to join this was also not only looking at actionable mutations from the point of view of choice of drugs, but you can also use that information as to when you don't want to choose some of the drugs, the resistance. The classical example is your KRAS resistance with regard to EGFR targeting. Now you also have STK11, which is associated with Peutz-Jeghers syndrome as a germline mutation, but STK11 mutation, somatic mutation, has been shown to be usually not responsive to your immune checkpoint inhibitors. So there are likely to be more and more of these kind of evidences coming up. And I think definitely this is one way of going about it, but please understand the kind of limitations which you have with this. I will finally conclude with just one analogy. I will say that CGP is something similar to your PET-CT. It gives a whole lot of information about the individual. I mean, you can identify the different sites, etc. But when you start off, there is also the possibility that you might mistake an inflammatory picture also for something different. So you need to bear that in mind. The CGP, again, has its own limitations. Limitations are the first thing which one needs to understand before you start using it on a routine basis. But I think I should thank Purvish for giving me this opportunity to be present and share some of my thoughts. But thank you again.
Very wonderfully said, sir. And I echo your thoughts. Sometimes I feel like a soothsayer or a astrologer reading Janam Patri, because there are so many different nuances and you don't know the surety. Aisa bhi ho sakta hai, waisa bhi ho sakta hai, ye bhi ho sakta hai, you know, all kinds of surety and unsurety. And that I feel is just because of the way this technology is developing. And so there are, of course, limitations that you have just outlined, and who better to come from than from you, a person who believes in this concept and still, and so the more we know a concept, the more we understand the limitations, and that's the feature of a wise man. And that's what you've outlined here, and true acceptance to that. I think we are in a field that is with all kinds of reservations, limitations, nuances. Having said that, these are the options available today for us to treat our patients. And in the world where it's
Dr. Sewanti, we can't hear you.
The image is also frozen. So while she is coming back, maybe we can take this opportunity to ask Dr. Aju to give his opinion and feedback on the discussion so far. Aju?
Yes, sir. Thank you. Let me also first, before we get into the discussion of the paper, recollect an incident from more than a decade ago when I was at ASCO. It was one of those IACA meetings, and I was a novice oncologist. And there I was in the outskirts of a mob of people mobbing the real hero of Indian oncology, Purvish sir. So at that time, I think I got a chance to just grab his hands and shake his hands. That's my earliest memory of meeting Purvish sir. But thank you for this opportunity to discuss this paper. Obviously, this is an awesome peer review. I'm glad none of you were peer reviewers for this manuscript, especially Purvish sir. At the outset, I must say I'm just a hobby researcher. I'm not, academician. So I do research if medical students come to me and say they need a project. So kudos to my medical student who's now a doctor, Dr. Serena, to get all this work done. Without her support, I wouldn't have been able to do this work. Now, all research starts with a question. The question that brought out this research was a real-world issue where I did not know how to give appropriate counseling to my patient before I order NGS. I do order NGS even now. So I did not know, what should I say? Will this provide a 50% chance of a clinically actionable mutation, 10%, 1%? What is the realistic number? There was no number from an Indian context. And considering the fact that a lot of us order very few, I decided to then crowdsource, and I posted on multiple WhatsApp groups whether anybody wanted to join me on this project. And about 11 other oncologists agreed to join, and Nirmal took the lead in that, Nirmal route. And thanks to all of my co-authors, we managed to put together a reasonable size of reports. Now, this is a real-world study. This is the first data from India. So essentially, my goal was not to get the purest data. My goal was to get the first data and be open to scientific criticism as is happening here, and await further publications and manuscript to refute or support the premise that has been described. Because of the retrospective nature of the paper, how to assess clinical utility was a big question. And if I'm made a host, I can share a manuscript on my screen. So please make me a host, and then I can help with that. So while I was actually debating in my mind, how can I how can I actually make a clinical utility answer, an endpoint? That is when a wonderful paper, which I would like to bring your attention to. Am I a host at this time? Let me try. Yeah, maybe. So, okay. So this paper at that point of time came into our public stream. That is the assessment of clinical benefit of integrative genomic profile in advanced tumors from University of Michigan, Arul Chinnayan's group. So they actually had about 1,138 patients on whom NGS was attempted, 1,115 who actually got successful NGS. And how did they describe the clinical utility? And this actually, helped me frame the endpoint for the study. So let me expand this. So the clinical benefit rate, which I mirrored in our manuscript, was defined as a proportion of patients receiving sequencing directed therapy for 6 months or longer. This is exactly what I have decided to use it. Obviously, there may be differences, like Purvish sir said, one oncologist, there is doctor shopping. So someone goes to an oncologist now here, and then that person goes to another oncologist. Will that be captured in this data? I don't know. So this is exactly how I defined exceptional responders received SDT for a duration of 12 months or longer. And what did they find? In this study, what they found was about 37% of patients had clinical benefit, 37%. Now, that is 49 out of, let's say, 1138 patients on whom the test was attempted. That is 37%. But, I then decided to do the calculation where, I wanted to give counseling to my patient when they come to me before I order the test and tell them, what are the chances there may be some benefit. So there's no point giving counseling on how many got benefit, how many got a treatment. So in discussion with my co-authors, all of them, we decided that we will actually use the denominator as the total number of reports, total number of patients on whom the test was done. And based on that, we came up with this number, which you quoted as 4%. So 4 out of 100 got some clinical benefit. Now, very interestingly, if you actually do that exact calculation in this manuscript, that is 49 over 1138, or for that matter, 49 over 1015. This is what they come up with. I don't know if you can see my calculator screen. Can you? Maybe not.
No.
It's essentially 4.8%. So the point I can tell you from this manuscript is, although I did say judicious use of NGS is necessary, shared decision making, I think that's the word I used, is important. What I gained from this paper, which I did not put in that manuscript because this was out of the purview, it was all about describing what we found and not about interpreting it. What I found was, the benefit of NGS remains the same in India versus the West. Okay, it's about 4%. Now that brings up another interesting question. We know that our outcomes in chemotherapy are different when compared to outcomes in the West. Patients may live longer in the West when compared to patients in India for various reasons. I don't know, maybe antibiotic resistance when they get infection, maybe lack of second-line therapy option. But if there is a targeted therapy option that is proven to be a tier 1 or proven to be efficacious, and actionable, then I think the benefit that patients in India will stand to be similar to that in the West, is what I can interpret from my analysis.
Thank you so much.
Yeah, that is how. Yeah. So that's my brief summary.
Conclusion. I just, as a moderator, I would like to point out a couple of things and then take it to the next panelist, because you said something that needs to be pondered upon. The benefit of NGS in Indian patients, 4%, and benefit of NGS in the Western patients, 4%. Does everyone think that way? And that answers this slide, actually. What is the definition of actionability, and how much, do you think we are benefiting from the testing? And, with that, I come to the panel asking, what do we think is NGS doing? What is NGS? Is NGS, is just 160 genes? Is NGS 400 gene profile? Is it DNA? Is it RNA? What is it? And so that was a very strong statement, 4%. So I think Dr. Parikh negated that statement right at the start, and I also stand in that team to really challenge that statement. And in terms of actionability, we have seen much larger actionability of therapy, of pathways in the novel trials that are coming up, or the novel approaches that are coming up. One such novel approach is in front of you here, where they are looking at small tumor samples and PCR-based CGP performance, and how the actionability can be enhanced to a larger extent by performing nuanced testing. And the reason to just bring up things like these is also that methods of capturing the pathways and then methods of treating may vary. One example is also multi-pathway therapy. A Biacon trial is an ardent example where BRAF V600E was treated with 3 drugs in context of colorectal cancer. And here, one drug would not have worked. So when we talk about actionability and results of actionability, we also need to take into context what else was there that could have been treated, and whether it was monotherapy we are talking about, whether we are thinking beyond monotherapy. And I'm going to show a couple of other slides and concepts, and I will ask some questions. So I know Amish is not in the panel and we are running short of time, but I will ask Amish what his thoughts are with regards to actionability in India, and what is the thought around what we have just heard from Dr. Aju Matthews regarding the 10 and the 4%. And I think here I would like you to shed some thoughts on monotherapy versus multi-pathway treatment that we spoke to Dr. Amit Verma with.
Sewanti, first of all, I stand anywhere in the queue where you stand, I'm behind you. So it's not only this 4%. I honestly don't agree with this paper's conclusion. Whether as on this platform, if I read Google, if I follow Dr. Amit Verma's advice, and if I follow Molecular Oncology Tumor Board, nowhere my personal experience, and I have 70 to 80 CGP reports at least in my file, it is close to 4%. One. Second most important thing, and I say this again and again, we really don't know how to read. Beacon trial is a classical example, Sewanti. BRAF in colon, you give encorafenib 2.6 months. You add cetuximab to that because you understand the pathway. You block BRAF, EGFR will be activated. If I don't know this, how am I going to treat patient? The important point, what I feel from this paper, is we all need to learn to read actionability, as you said. So to answer your question, I do not understand actionability even 50%. And that's the reason probably we are failing in helping our patients. We have the technology available, but we don't have the knowledge available.
Very well said. Thank you. So moving on to a couple of studies, and I think I may have a few minutes, sir. Few minutes now to complete the moderation.
Please continue. Please continue.
So I bring to the front the I Predict manuscripts, 2 publications by Ray Kurzrock and her team, Shumi Kato being one of the leaders in this. And you look at how they programmed the profiling. They figured out a particular, so they did DNA-based profiling. They included broad panel sequences. They included molecular tumor board understanding and interpretation of their reports. They had tumor tissue, they had circulating tumor DNA, so liquid biopsy based testing, and they've developed something that was very unique, and that was a score. They developed a score that you are seeing here, and based on the scoring, matching score, they then gave the patients their treatment. They sat down and they figured out most of these patients actually received 2 drugs together. And if you see here, if the matching score, which they calculated by the number of pathways seen, by the number of pathways targeted divided by the number of pathways seen, they showed actually that if the matching score was above 50%, these patients lived longer, they have higher actionability, and they had higher progression-free survival. The other paper based on the same study, this is a follow-up paper that was published later, again gives you the follow-up data for this study where matching score now 60%, less than 60%, unmatched, matched, and what was the CR and PR rates, and you have it all outlined here. And I show this only because this is yet another example for practicing precision CGP-based treatment, and in context of the paper that we are discussing, very different results, very different actionability that we saw based on iPredict. One of the major questions I had here, and for which I will bring it back to the panel, and a question to Dr. Nikhil, what did you think of the exclusion of these patients from the from the main cohort that was analyzed, and the title of the paper.
So again, coming back to the list of the mutations, I would say genomic aberrations, which were excluded from the study, for the understanding that this particular study was done to, I mean, rather the authors probably wanted to really fortify the patients who actually had certain uncommon mutations, and that was probably one of the reasons. In the day-to-day practice, this is not something what we do. There are occasions where we also had seen, let us say, the EGFR might be negative by PCR, but we might have picked up the same through NGS. So that has been the case. So although we do understand there are certain mutations which can be picked up by a simple study or more like a hotspot mutation technique, but in the NGS, probably I would still like to see the entire, all the reports in toto. And again, there are times when we come across multiple aberrations, genetic aberrations, and that makes our life extremely difficult, how to really interpret what to do with those tests. So probably I'm not too sure why these particular numbers were excluded, because that would have really increased the numbers, increase the number of actionable mutations. And probably that is something what we are doing in day-to-day practice.
Great. So I felt the same. I felt that the exclusion should not have been made, mainly because irrespective of what was the method of testing, essentially these are mutations that are targetable pathways. And by excluding them, the title becomes misleading, as Parikh sir had said in his interview. Coming to this concept of 297 CGPs with 12 oncologists, the number is 24 tests per year per oncologist. I'm going to turn to Dr. Vamsi. Vamsi, what do you think of this number? Are you doing 2 CGPs a month?
I think this number is probably higher now. I must be doing 4 or 5. I don't think I'm still going to probably 30, 50 per person. And again, you know, it's good to have some balance. I think it's nice, like I said, academically you would want to do in a lot of patients, but where you are having a disease which is very aggressive, where your performance status is not great, where affordability is an issue, I will not ask for a CGP in such patients, because even if I get a drug that might be an actionable thing, it is not going to be possible to give it to the patient. So I think that is very important. It's not the fault of the CGP. It's just the fault. It's the way the situation is. A patient cannot go for the whole hassle of getting the drug from abroad. Then why should I waste money doing the CGP in the first place? So in that aspect, I agree with Aju that these results are in a way good. The opposite sense is that it's always good that if you get a result and the patient is willing to go for the extra mile, it's worth doing a CGP. And I would order it for practically every patient in that case. But as a balance, in my practice, it's probably 4 or 5 in a month. I would offer it for under 10 more, but I think that's about the number which do it every year, in my practice at least.
Excellent. Thank you. So you actually addressed the barriers to testing as well, where affordability is one of our big issues. I agree, where you know that the applicability is a question. You really don't want to do a test where you can't figure out what's to be done with the results.
So other thing, you know, this last point of yours, raising awareness for testing, I think that's a bit of a misleading thing, because it's not that most medical oncologists are unaware of this. You know, somebody who is sitting in a center which sees 90% government or a reimbursable sector would not be able to do this. It doesn't mean they're unaware. You know, by making this statement, it's kind of a derogatory statement saying that, oh, somebody sits there, he doesn't know the test, so he's not ordering it. That's not the case. I think most oncologists in India are well trained.
Yeah. Thank you for pointing that out. I agree. I completely agree that it comes from actually a different mindset, that most have not gone into it because they have not found it meaningful enough or feasible enough. You know, what do they do after the testing? And I'm from Patna, and at one time I asked, why wasn't EGFR testing being done? And the answer was because there are no drugs. And so I totally get it. Probably should have worded this differently in terms of raising awareness for feasibility to testing. You know, where are the feasibilities? Where can we get it for free? Where can we get the drugs for free? What is the compassionate access availability? Whether clinical trials available? I think those would be more important to put forward and not raising awareness. Maybe raising awareness in terms of patients, because patients also deny many times to go through biopsy. There's a lot of misinformation regarding biopsies and how biopsies could lead to increment in cancer, and repeat biopsies are a big taboo. And so I think raising awareness around that, I feel, which I call precision biopsy, I think is much more applicable. So I will skip this because I've asked most people about the actionability at their center, or how do they view it. Coming to a very salient and one of the last points of molecular tumor board, I come to Dr. Randeep. Randeep, your thoughts on molecular tumor board, and then Vineet can take the next question. What you guys feel about molecular tumor board, and what is the role of molecular tumor board? Do you find it useful, or how do you discuss your cases?
So molecular tumor board is indispensable. You may not have it at a local level, but you can join hands with colleagues and discuss it. Every case where you have a mutation, there is no need to take it to molecular tumor board. If I have a clear-cut case of an EGFR mutation on lung cancer and there is no mutation, there is no point in taking that to the molecular tumor board. If I have a IDH1 mutation and I don't have the access to the drug, there is no point in taking a cholangio to molecular tumor board. But yes, if you have some rare mutation, if I feel that this is actionable and the lab feels this is not actionable, if I feel that this is not actionable, lab feels it is actionable, if there are double mutations, if there are some rare mutations, if there is polymorphism, if there is a issue about VAF, I think, yes, it is helpful. In my case, usually I send the cases to Dr. Bhavna, where she conducts the molecular tumor board. And I think 25% of the cases go there where I'm stuck. Where I'm not stuck, I try to sort it out first myself, then discuss with the lab, then check some literature. And then if I feel, yes, I'm not yet satisfied to prescribe the drug and I'm not myself convinced, then I take it to the molecular tumor board. So out of 100, around 20 reports may go to molecular tumor board.
Excellent. Thank you. Thank you. I think that's the number, that's the kind of number we are dealing with in India. I have also personally felt the limitation of being able to bring, what the concept that is explained in the West, bring every case to the discussion in the molecular tumor board. I've felt limited because you cannot rally up all the cases where you have sort of simplified answers. You may be going wrong, but it's just not humanly possible to bring in all the cases to the discussion.
Sewanti, the case is that I don't have a tumor board in my institute. If I had a molecular tumor board in my, like a tumor board we have in our case, we discuss every case in the tumor board. Because I'm not having a molecular tumor board access directly under the roof of my institute, and we all have to rely, because we are not having that much patients. If I have a molecular oncologist and I have a molecular pathologist and all of us are there, we would like to meet every month and discuss every case. And if you have in a high volume center or an institute, the message is not that don't bring that case. This is what is the real world I do, and that is what I told you.
Excellent. Excellent. I think these are the real world limitations, and one of the reasons to bring this point forward is also because when we practice CGP and the data that gets spit out, and one of the messages I read from Dr. Parikh sir was also regarding raw data and the curation of the raw data and the accessibility to the raw data and interpretation of that. Who's going to do that interpretation? We are really short of people, of how to, it has been a challenge, and I can't tell you enough, and Amit stands witness to this. Whenever I have run MTBs, I've gone pleading to the molecular team, please, 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 who can champion today's meeting? Who can really be there to add to each case? Because it needs a lot of homework for even the molecular scientists to figure it out, curate it out, and then come and be able to add meaningfully to that particular case. Amit, any thoughts? And then Dr. Anuradha, I think we are winding up now. I will quickly present a two-liner on this case that I have here, scoot over the next cases, and then wrap up with some last messages from the panelists. So Amit, your thoughts on the molecular tumor board?
I think it's a must, even if you don't feel that you're not going to learn in that tumor board discussion, but there's always something to learn. Recently, I had a discussion on the molecular tumor board where I learned that a TP53 mutation R273C is a one which is called gain of function mutation, where PARP inhibitor can also be functional. So these are the things which can come up because the knowledge is advancing so much, and you cannot keep pace up with all the time. So your colleague can teach you better than the books actually. So with that, MTB is must there.
Thank you. And I feel that it's an ever learning scenario, and application to therapy changes when you discuss cases in forums that are deliberating on TP. I will quickly show a couple of pointers on the cases. So this is a 69-year-old who had a history of breast cancer, who presented with something that was labeled as triple negative breast cancer and was PD-L1 positive. Some of you must have seen this case presentation earlier. So this was when she had breast cancer. This was when she presented to us. Multiple nodes here you can see. And this is the subcutaneous nodule, read as poorly differentiated carcinoma, ER/PR HER2 negative, called triple negative breast cancer, was having GI bleed. Got endoscopy done. She was presented to me as triple negative, but we got the endoscopy done. Turned out to be a gastric cancer. And this is the profile. She had a sister who died of breast cancer. She was BRCA2, and she was MSI high and a TMB high. And then there were so many alterations, and we had to sit down and figure out whether the alterations were coming, whether the MSI was coming from BRCA, whether it was coming from the BRAF, the atypical BRAF, or it was coming from her own history of Lynch. And there was also CPS positivity, which came back later. And a lot of thoughts that revolved around the discussion that we had, whether this is HBOC, whether this is Lynch, whether we treat her with chemoimmuno, whether we give her PARP inhibitor, whether we treat BRAF, whether we look at MSI, TMB, do you do combination treatment? Everything simplified, still treated with frontline treatment with checkmate 649, and the results are in front of you. The patient remains disease-free on pembrolizumab nivolumab monotherapy as maintenance. And I have presented this case to different varied forums, and the thoughts are that the BRCA positivity makes her MSI high, and irrespective of what the MSI high status comes from, the patient will still continue to respond to immunotherapy, and that's why the patient has been kept on monotherapy with immunotherapy, no PARP added. And this is an advanced prostate cancer where BRCA2 was found in a liquid biopsy, and this was then read in one molecular tumor board, read as pathogenic. Patient was put on olaparib, and in another molecular tumor board, it was read as CHIP. And you can see the curve. This patient is a doctor. He has drawn his own curves. And you can see that it went down with other treatment, but then it's coming back up. And truly, the challenge is whether this is just a CHIP. And if you ask me today, it is a CHIP because his PSA levels are coming back up. I'll scoot over the third case, and I'll leave you with my last questions. So when you look at the iPredict, the gospel today we have are the two manuscripts from iPredict that really are sort of leading the discussions in precision oncology. And the message is for discussion forums. So the reports are getting so difficult to understand today that none of us know it all. And that's where internationally also people are applying this method of curation, where people sit down and curate the data together. There's a lot of AI-based mechanism coming in. We know about cell search. We know about there's a Ray Kurzrock company that I'm blanking out on, and many, many more AI-based methods that are coming through. So MTB in iPredict, it talks about the MTB that they did weekly. They also did an emergency MTB called just-in-time MTB, and then they did virtual MTBs called track, and then they talk about a real-world MTB with an N-of-1 approach for patients. It is just not possible in India. You cannot have N-of-1 approach for millions and billions of patients that we have here today. And this is my last slide before I go here. Final words from Parikh sir. Coming back to you for a closure, sir. What do you think of this slide and the number of options we have today, and the concept of CGP and the data it spits out for us to toy around with, and the multi-pathway therapies that are now emerging?
So absolutely, Sewanti, I agree with you that we are now spoiled for choice. And the only limitation is whether the patient can afford the treatment that is intended or suggested by the molecular test. As long as the patient can afford, and it is, I must say that in the state of Rajasthan, all anti-cancer treatment and all anti-cancer diagnosis is free, provided by the government. So the cost factor discussion gets cut off when we are at Rajasthan. But I think, thank you very much. You have done a fantastic job of moderating this panel. And with your permission, we'll move to the final conclusion by our Amish. Please go ahead.
Yes, sir. Just before, because Dr. Sewanti asked all the questions, she didn't answer some of the questions. The two cases which you presented, Sewanti, the first one was, there are plethora of mutations observed. So normally in such case, my phone call will go to Dr. Amit, and he will say, okay, you give nivolumab because BRCA is this thing. Then I'll read Google and I'll tell him CCND1 has been shown to have immunoresistance, and TERT mutation has shown to have action of ipilimumab. Why shouldn't I not give Nivo plus EP? So that is a question. Normally I ask him, he knows about all these things, and I don't think I have an answer. The second is very interesting, the Sewanti, the doctor himself, rather than CHIP, I personally think is that the PARP inhibitor, the moment you block that, there are the other pathways which are coming up. And I think Beacon has shown us that, the world is open. So the last slide, what you have shown, probably we need to have juggling of these drugs in a way that the way we look at TPF, that is docetaxel, cisplatin, and 5-FU, similarly, I think we will have ramucirumab, erlotinib, and pembrolizumab, something like that. So my question were to you, your thoughts about this. But if you don't want to answer, I will tell Amit to include that in his concluding remarks.
So, Amit, we must go to Amit for his conclusions as well. But I will give you my quick 2 cents. I was baffled by these two reports. Both the patients are so-called very, very important people who had approached me through others, and I needed to know what I was doing. And truly, even I was completely baffled by the kind of reports that came in. And when I enlist those are a reality. Until date, when I took this case to the GEM meeting last year, my gastric case was selected for the GEM presentation last year at the Foundation 1 meeting. They also, the jury was out whether it should be PARP or it should be immunotherapy. But Amit is well aware of both the cases. I presented them at every forum. So I presented them at Impact also to try and understand, what are their thoughts. And, we went with a simplistic approach because I was reassured by the molecular experts that the BRCA was contributing to the MSI high. And then we looked up a lot of different literature where what is the MSI high from? Does that determine anything, or is it just the MSI high status? And really, honestly, unless it is BRAF, it is the MSI high status that drives the response from immunotherapy. And that's how that action was taken, that risk was taken. But your comment about the second patient, the doctor, is very well taken. He was germline negative, and the BRCA2 in the report was, and just as Amit said, a low VAF doesn't mean anything in the liquid biopsy. He had a very low VAF and really didn't know how to interpret. By the time I said no to olaparib, he had gone to New York, and William O had started him on olaparib, and the germline was negative, and his daughter was also germline negative. She had breast cancer. And so really everything up in the air now. The patient himself sends me that he's no longer responding to olaparib. And that was an eye-opener where everybody had contemplated CHIP, but we didn't have the means to do the WBC testing to figure out whether this was CHIP or not CHIP. So bringing it forward to Dr. Amit Verma for his final comments. But Amit, just before I leave it on to you, just a last observation that I read through many precision manuscripts that were there, where I read in comparison with Aju's manuscript. And PFS2 remains one of the biggest outcome measures, where you measure the patient's own PFS1 and then whether the last therapy and how long the last therapy lasted versus this therapy, and above 1.3 times of PFS2 advantage is considered meaningful enough. So Aju showed us another outcome measure that was very interesting to see, and that he had followed, but mostly precision trials have followed PFS2, and I leave you with that message and bring it to Amit, the guru, for his work.
Dr. Sewanti, it is my job to introduce Amit and not yours.
Sorry. I don't want to take away.
I'm just joking.
I'll reserve my introduction.
No, I think I met Amit for the first time in Max Hospital, and a simple looking guy in a very polite mannerism. My first thought process was, kahan se aa gaya yaar? At least, very polite mannerism. But when he starts speaking of science, you feel, my God, itne dimag mein kya bhara hua hai. Amit, he has done his molecular oncology from MD Anderson, and I think there is not a single medical oncologist in India who has not even called him once. And he's always ready to answer. And he, in fact, if he finds it interesting, after 2 months, he will remember to call you and say, ki wo patient ka kya hua. Like, I can say for sure that 80 to 90% of my superb responders of CGP, all because of Dr. Amit Verma. And there cannot be anybody other than Amit who can, give a final conclusion. अब सेवंती की जैसा मतलब मुझे भी चुप होना है. So Amit, you can speak, and 10 minutes are yours.
Thank you very much, Amish. मैं आपको पार्टी दूंगा बाद में. Okay, so can I share my screen, please?
Yes, please.
Imanchi, can you make?
Can you see my screen now?
Yes.
Okay, great. So I think the moderation was great. Dr. Amish did a great job interviewing Dr. Parikh, and I totally agree whatever has been discussed in the whole forum. And for this particular case, I will give my personal thoughts on it, and then we can conclude. So the outline of the study and the conclusion is very well discussed. Here, the impression was that the CGP is only limited to the target therapy. I have just highlighted that, and we have discussed the 10% and the 4% through this whole discussion. So what I found that the observations from this study is that it's a first time real world experience coming from India, a multi-institutional for sure, a very positive step, which gives an indication that the comprehensive genomic profiling is getting into the mainstream of oncology practice, which I'm very happy to see. More and more doctors are looking to do CGP to inform their treatment decisions. So again, very happy to see that. Interestingly, what I saw that many different cancers beyond breast cancer and the lung cancer, all were subjected to CGP. So it's a true tumor agnostic approach which people have applied. And also I've seen there are the treatment lines from 0 to 5 are the criteria, which reflects that the upfront usage of CGP is also coming up. But the downside of the whole paper was that the authors have underplayed the potential of both the study as well as CGP as a technology, which I'm going to defend now with few slides here. So the key objective of the paper was to study the clinical benefit of comprehensive genome profiling in advanced cancers in India. But unfortunately, a significant proportion of the patients were excluded. The exclusion criteria states that genetic alterations that have well-validated standardized target therapy were excluded. So it's a big question why they were excluded. Though they have explained it's an established thing, but again, the guidelines also talks about CGP as one of the methods to identify the targets or the actionability in these cases as well. So this arbitrary inclusion criteria led to 76 cases with treatable variants to be removed, leading to a significant reduced number of cases to be benefited from CGP. If we are to include these cases, 76 cases increase the percent of the patient receiving target therapy to 97 in total, that is 32%. So whatever is represented 10% is actually 32% if I were to write the paper actually. This also reflected that the authors believe the use of CGP only in second line or third line setting because the first line standard of care were always taken into account while the cases where the CGP was not used taking the decisions. Let's talk about the second important aspect, the clinical actionability, which was discussed in a single liner. It was not discussed anywhere in the whole paper. 96 patients, that is 43%, had target therapy option in the form of approved drug. Very interesting number. But actual pickup rate, which is reported, is not discussed in the conclusion at all, which is trying to underplay the whole gist of the CGP. If I were to recalculate the entire 297 cases, the pickup rate is practically the 96 plus the 76 which were excluded, that is 57.9%. That is close to 60%. That is something very important for us to take home. The drawback, there was no discussion on the biomarker used for immunotherapy, TMB or MSI, or tumor agnostic biomarker like NTRK, or LOH score or HRD score or HRR gene, which reflects that either these technologies were not coming up or giving the information in toto, nor it were analyzed. And this is going to give us a small fall in the clinical actionability. Had we had this information, the clinical actionability might have gone up. So if we try to see the different commercial genetic tests considered in the same yardstick in this case, out of the 14 tests used, 4 of the tests have been published in a robust manner, where clinical validity as well as analytical validity have been done. Out of 14 tests, 5 are only hotspot tests. So in a comprehensive genomic profiling where we do end-to-end analysis of the exon as well as the intron exon boundary, these hotspot assays are not to be used or are misleading when we compare the CGP with these hotspots. So out of 14, 5 were the ones which were not appropriately chosen to include in the analysis. Tumor agnostic biomarkers like NTRK, MSI, and TMB were not tested in these 5 basic hotspot assays, which we know. So these false negative results which can come can impact the overall pickup rate of the CGP in this whole paper. Let us talk about the reasons for not getting the genetic testing based target therapy. The 21% and plus the 22.6%, close to 24% patients were not given therapy based because the patient still was receiving standard of care or off-label options were not available, or disease under remission or indication for therapy. This reflects that the CGP can still be used in these patients for in future. Again, bringing up the idea that yes, CGP still is an option and the percentage what we are claiming is relatively low. Let us talk about the definition of clinical benefit. This is a paper, this is a slide which I have taken from the FDA. It categorically mentions that clinical benefit is favorable effect of any meaningful aspect of how a patient feels, functions, or survives as a result of a treatment. So it is talking about the intervention. So the clinical benefit, which as per the paper analysis says that 17 patients were given the treatment out of 217 and 9 showed the response, hence the number is 4%. If I were to calculate the data based on this definition, ideal would be the 9 patients who showed the clinical benefit out of the 17 patients who were given the treatment, that comes out to be 52.9%. So the actual clinical benefit is more than 50%. Again, the response criteria of 6 months have been discussed earlier, and the ideal would be the PFS ratio, PFS2 by PFS1 more than 1.3% as discussed by Sewanti. So this ideal or more established, robust way of response was not evaluated. Again, giving us a little bit of lack of confidence in the analysis. If we talk about the clinical utility, it's a definition coming from the NCI, which says that the term that refers to likelihood that a test will, by promoting an intervention, results in an improved health outcome. The clinical utility of a genetic test is based on the health benefit related to the interventions offered to the individual with positive test results. So if you see the clinical utility, the patients, 9 patients who were given the benefit, plus 76 patients who were already, as per the standard guidelines, who were picked up on the CGP, total were 85. So if we divide the 85 by 297, the total cohort, the clinical utility is in the range of 30%. So again, we are trying to underplay with the data, which is practically a little higher. To summarize, this is a retrospective analysis. We have just witnessed the underplaying of the data. The study is limited to the utility of CGP in target therapies only, not in the immunotherapy or in other chemotherapy, where this genomic information sometimes guides you the chemotherapy options as well. If you see the patients, 21 patients who were treated based on the genomic therapy, 12 were either EGFR or HER2-based treatment approaches, which reflects that the physician who were treating the patient were not comfortable going beyond the few pathways which were observed in the other categories. Monotherapy or single pathway driven therapeutic approach was applied while we were trying to see what was the treatment approach given to the patient who responded well. To talk about the future, I would expect that a prospective trial is must, again multi-institutional. The treatment option should be MTB directed therapy rather than an individual approach therapy, so that we can have a uniform platform to evaluate the responses. A monotherapy eventually has to fade away. A combinatorial approach is must. And then we should have a better response evaluation criteria, which has to be individualized. With that, I thank you all for your kind attention. And over to Dr. Amish and Dr. Parikh, sir.
Amit, as always, amazing, pertinent questions. Whenever you speak, na, I feel like a paper pen leke likhna shuru kar do, kal bhul jaunga wapas karke. So the one important question, Parikh sir, we are supposed to conclude, but I can't resist asking Amit one question. You talked about the clinical utility of NGS report. Suppose, say, ballpark figure is, say, 25%, assuming 1 in 4 respond. If you add the pathways which tell you not to use that particular drug, for example, don't use cetuximab, don't use immunotherapy, or don't use alpelisib, what would be the utility based on your clinical experience? And this maybe Sewanti also can take, because she's running a tumor board, so she also might have some clue towards that, because I believe this is extremely important, what not to use also. Dr. Raj also alluded to the same in the panel.
So CGP is truly a Janam Patri. In my experience, the number is 80% and above that, always. You talk about target therapy, you talk about immunotherapy, you talk about the resistance mechanism, you talk about the prognosis, you talk about the choice of chemotherapy, you talk about the hormonal therapy choices. So all the combination, all these things can be easily interpreted if we just try to see the context. There's a case in this particular paper where they are talking about in a uterine cancer, the patient was given everolimus, and it was a PIK3CA mutation observed. So I wanted to ask whether the everolimus was given as a monotherapy or was it given along with the hormonal therapy? Because the PIK3 mutation is causing resistance in the uterine cancer. And if you are not using hormonal therapy, then what is the drug who is truly responding to the cancer actually. So those nitty-gritties has to be taken into account while we do the interpretations.
Right. So Sewanti, last word, and then over to Parikh sir. Dr. Ajju Matthews' paper 4%, my take 25%. Amit says 80%. Now where are you standing, Sewanti?
So I'm somewhere in between. I think, Amish, the field is evolving.
Yes.
The field is evolving. We have to take it with a pinch of salt. We have to take it with the limitations that Dr. Rajkumar sir mentioned. Accepting all the limitations, I still feel this is the way to be. There is no other way. We are in an imperfect world, and we have to treat our patients with the imperfect science that we have. But rather than letting the science perish, this is the way to be. So thank you for the opportunity and for
Parikh sir, over to Parikh.
honor. Thank you. This was brilliant. I can't tell you how brilliant. Thank you.
One factor that we have not discussed is how the molecular alterations can affect the enzymes that metabolize the drugs.
Correct.
For instance, if there is irinotecan that I'm using for colorectal cancer, maybe there will be heterogeneity in STAR28 and the way that the drug is metabolized, and that can also indicate whether the patient should receive full dose or half dose or 25% reduction. And if you add that to 80% of Amit Verma, then it will be close to 90%. So with that, I really thank you, Amish, for not pulling my leg so much. I thank Sewanti for doing a fantastic job of moderating and sharing her such wonderful specific cases, which are so insightful. And I think Amit was like the last 3 overs in a T20 match. He made sure that he took the entire discussion to the next level. And I think everybody who waited till the end has immensely benefited from the slog overs that Amit Verma was hitting. So with that, I think I will remind you that again, second Tuesday of next month, we will have Oncology Beyond the Obvious. And till that time, please continue to give us feedback and also tell us how we can improve our session. Thank you very much.
Thank you. I think we can close the session now.
Right.
Thank you, sir.
Himanshi, Rahul, we can close the meeting.
Okay, sir.